I'm one of the ecologists at Watershed Center that studies not fish, although Peter Moyle would say frogs are just fish food. So anyway, I'm going to talk about some of my dissertation research, and I think what's really interesting is that these frogs in particular are really good indicators for fish and bugs and a lot of other things. So first off, I want to acknowledge one of the reasons why I get to do what I do, and my advisor Mike Miller, our lab director Sean, the Center for Watershed Sciences, which has been a great place to work, and really enjoy all the colleagues that I get to talk with every day. Brad Schaefer, Amy Lind, and many field helpers and folks that have been out in the river with me sampling. And then just a quick note, one of the reasons sort of that we do what we do is various, but for me, that's my son, and getting to go out in the field and, you know, have him catch a frog for the first time is sort of one of the best moments that I could ask for. So I really enjoy what I do, and I want to make sure that everybody else has that opportunity as well. So that's why I work with frogs. So California rivers, as we know, and rivers in general, are largely in rough shape. Freshwater species in North America have projected extinction rates that are 5 times higher than terrestrial animals. And there's quite a history of landscape change in rivers in California. And that really started with the legacy of hydraulic mining, gold mining, that started in the 1850s. And what's pretty incredible is that it literally transformed the landscape. I mean, we hosed hillsides down throughout the Sierras that permanently changed the geomorphology of these rivers and these landscapes. Just to give you a sense of how much sediment and how much landscape change that is, it's estimated that over 8 times more material was excavated from just 3 watersheds, the Yuba, Bear, and American, than during the entire construction of the Panama Canal. So all that sediment is just sort of wiped off the landscape and pushed out either into the bay or onto where we are now sitting to some degree. So that's an amazing amount of change. And then that lasted till about the 1900s or so. And then we have also dammed rivers throughout California. There are a lot of dams. California has probably is the state with one of the highest number of dams. And in addition to that, hydropower generation is a big part of the state. About 15% of our power generation comes from hydropower. And with residential energy demands, that's a really key piece of our infrastructure. So despite all this, this frog has persisted, which is pretty amazing. They're one of our only river breeding frogs, so they really only require moving water, and unfortunately they use this really wide range of habitat, from really small creeks to very large rivers, but they've disappeared from about over 50% of their historical range. And their range existed from all the way down to Southern California up into Oregon, and both on the coast and in the Sierra Nevadas. And so currently they're being evaluated as a candidate for listing both at the state and federal level under the Endangered Species Act. So despite all these crazy changes, frogs are still here. They're still around in a lot of places. This is a small tributary, small creek in North Fork American watershed. This is the North Fork American River, which is one of the only undammed, unregulated rivers in the northern Sierras. And unfortunately, they're barely holding on in a lot of places like this. This is a diversion dam on Slate Creek in the North Yuba watershed. And believe it or not, they actually breed right below that dam in most years. But you can imagine when a spill comes through, it can completely scour those eggs out. So I'll talk about the biology in a second. So these frogs are amazing indicators of hydrology, and I say that because they're really strongly linked to the local hydrology and thus really the watershed history, and that history is pretty pretty extensive, as you can see. Mining, dams, all these different things that we've done. And that's because spawning timing and habitat selection is really tied to these receding flow cues and increasing water temperatures. So these frogs in the Sierras really key in on the snowmelt recession, sort of as the snow is melting, the flows are dropping, and there's this very amazing consistent pattern that occurs throughout the Sierras. On the coast, it's more of a rain-driven signal, but it's the same concept. And just to give you a sense, over 90% of all the eggs we've seen fall within this very strict window of sort of less than 0.6 meters and depth and 0.2 meters per second or something like that. So, or 0.1, 0.2 meters per second. So they have this very amazing ability to select a very specific habitat, no matter where they are, and that correlates with both the timing and space. So they make a really good indicator of a river's hydrology. So my research is really trying to figure out how does flow regulation, and in particular I'm going to talk mostly about hydropower flow regulation, how does that affect the genetic patterns of these species? And so when I say genetic patterns, I'm really talking about fragmentation and isolation. And more specifically, can we quantify that genetic signature with specific hydrologic metrics of flow impairment? So a lot of the things we're talking about yesterday and today relate to metrics. How can we quantify metrics that we can use in management for environmental flows or healthy rivers, et cetera? And so I'm using a method that I'm not going to talk much about because I don't want to put people to sleep. But genome-wide methods are pretty incredible, and we're using something called RAD-seq and Rapture. And these are really powerful methods to look at genome-wide variation across the genome of really any organism. So we can use this with fish, with frogs, bugs. It's really powerful, and it's a pretty incredible method that we can use. So I'm going to start the study area that I've been focusing on, or that I'm going to focus on today. I have samples from all over the state and also in Oregon, but largely I'm going to focus on the northern Sierras. So the North Yuba, the Bear River is kind of squished in between there and is not shown on the map, but that's included in these results I'm going to show, and the North Fork American. And these it makes a really good comparison because both the North Yuba and the North Fork American are unregulated, undammed rivers, or at least above the lowest rim dam, they're undammed and unregulated. But that's pretty much it in the northern Sierras for undammed rivers. And then these other rivers have various regulation patterns. And so if we zoom in a little bit, let's just look at the American watershed. All those dots are just sampling sites. So I've walked through all kinds of rivers and into all these tributaries to sample frogs. And so on the main stem river on the Middle Fork American, in red down on the bottom there are no frogs in the main stem river. They're only in the tributaries, and that's because that's a hydro peaking. It goes up and down every day. I'll show a hydrograph in a second. Versus the North Fork American, I have samples going all the way up to pretty much the head of the watershed. Throughout, the frogs occur through the entire watershed. And the Rubicon there is a bypass reach, so they divert water out of the Rubicon to generate power, and then it flows back into the Middle Fork American. Just to give you a sense, so there's these really complicated flow patterns and river regulation that occurs, but I have samples throughout these. So, flow patterns. This is an unimpaired river. This is the North Fork American. You can see over multiple years, this is a log scale just because we get some really big flows that flow through here. But you can see the pattern is pretty consistent year after year after year. And what's amazing is that it can shift as much as 2 months in any given year. In a drought year, it can be 2 months earlier versus last year it was quite late. But the frogs consistently will breed right during that sort of nice descending limb of the hydrograph that is the recession limb, if you want to call it that. And so that, as compared to a regulated river, and this is an extreme example, but this is the hydro peaking reach on the Middle Fork American, and you can see every day when they start generating power, the flows go up from about 100 CFS to 1,000, and that's about a meter of flow change. And so that's pretty incredible. I don't know why that mouse is dancing around there, sorry. Oh, here we go, that's why. Perfect, all right. So you can imagine if you're a frog trying to breed in there, which they do on occasion, the problem is the flows will drop out from under after they've laid an egg mass, or it'll come up and it'll scour that egg, or in this case, desiccate it completely. So you can't really see, but there's a bunch of little black pepper spots in there that are little desiccated tadpoles or eggs. So results, so we basically looked at samples across the Sierra Nevada, and then I'm using principal component analysis, a PCA, to just look at structure. So across all these markers, genetic markers that I have, there's thousands, so we're looking at like 7,500 markers, genome-wide. What does the pattern look like? And most of the samples kind of break out by watershed, and so you can see that you know, the North Fork American is this big blue triangles, so it's unregulated triangles. But what's interesting is the Middle Fork American is really kind of spread out in two different groups. So that's surprising. You would expect things to sort of fall out by watershed just based on the species history. And so if we zoom in and look at the structure, you'll notice there's very little structure in the unimpaired watershed. And structure is important because it tells us a lot about population size, connectivity, how isolated they are. With a lot of structure, you'd expect populations to be fairly isolated. They're not moving around much. You might have less genetic variation. So in the North Fork American, they are moving around a lot. So really high connectivity, not much structure. And this includes samples that are, you know, 30 to 40 river kilometers apart, and yet you're seeing a really nice spread. Compare that to the Middle Fork American, and you'll see that you've got really high structure, implying that these are fairly isolated individuals or individual populations, and that they aren't moving around much. So another way to look at this is to use a metric called FST, which is just a measure of population differentiation based due to genetic structure. So how similar are in populations versus others, and it's scaled from 0 to 1. So 0, they're completely mixed, 1, they're completely different. And if I basically take the mean FST between all the different pairs within the watershed, and then you can see this by regulated, so if it's in a regulated watershed or not. There's a pretty striking difference. So there's a much higher elevated FST in the regulated watersheds than there is in the unregulated or unimpaired watersheds. And this is scaled against river distance on the bottom. You would expect the pattern of FST is that as populations are further apart, they're going to be more isolated. It's harder to get to from one population to another. So the trend is what you would expect. And the blue line on the bottom is kind of what genetic theory would suggest. The red line above there is really striking, and it's a bit concerning because these dams and these flow regimes haven't been in that long. So we're talking since 1950-ish. Generation time for these frogs is maybe 5 years-ish. We know they can live beyond that, but in a very short period of time, we're seeing a genetic signature of these flow regimes. And if we look at stream order, so this is FST versus stream order, and stream order is independent of genetic distance, right? So smaller watersheds or headwater streams have a stream order of like 1. So on the left, the small tributaries are on the far left, and the main stem rivers are on the far right. The pattern is opposite. And so this again, on the bottom, the unimpaired river is the trend you would expect, where the small tributaries are further away, typically, and more isolated, so they would have a higher FST, whereas the main stem rivers are more connected, populations can move around more easily, potentially, and that pattern is opposite in the main stem of the regulated river, and that's because most of the regulation doesn't occur in the tributaries, it occurs in the main stem river, right? So think of the main stem rivers as like the highway that these frogs are moving around through to get from one tributary to another, or even just using the main stem river, and so we're seeing higher FSTs in the main stem river, which really implies that regulation is really the direct cause. So one other thing we look at is genomic variation, so genetic diversity. And one of the things you can do is you can calculate diversity in a couple different ways, and by differencing those things, you can then look at the trajectory of a population. So is it losing diversity? Is it gaining diversity? That tells us a lot about the population health, the genetic health of the population. And so that blue dashed line, if things are around that line or below that line, they're expanding. If they're above that line, they're bottlenecking or losing diversity. All of the populations in the regulated watersheds are losing diversity. A few in the unimpaired watersheds are losing diversity, but the bulk are actually not. They're sort of neutral or expanding. And these are significant between the two, but I think the main thing is the pattern is what I think is concerning, is that not only are these populations isolated, they're also losing diversity. They're small populations, they have limited connectivity, so if one does sort of become extirpated or blip out, the chances that they're going to recover are pretty slim. So finally, one of the cool things we can do now is, are these metrics of flow the strongest factor in these patterns? So I can now actually model specific flow metrics from all the cool stuff everybody's been talking about. We can actually put these into these models against these genomic data and say, like, which ones fall out as most important. So which ones might be good indicators of genomic health. And this, and particularly in environmental flow, is a pretty neat way to integrate things. So this is just a really quick example. So I'm in the process of running these models, working with folks, Ted Grantham, Sarah Yarnell, Eric Stein. A bunch of people are working on environmental flows in California. And so it's really exciting that we might be able to actually integrate some of this genomic data for a frog that's very hydrologically sensitive into something that actually has direct management implications. So this model is boosted regression tree model, which is very similar to the random forests that folks have talked about if you're familiar with those. But the thing that I wanted to point out is this is just a model of, so what is the relative influence of any of these given variables on FST, that measure of isolation? And regulation, and I've run this with various metrics of regulation, so flow patterns, seasonality, predictability. In every case, flow patterns come out as the strongest predictor of FST over distance. That's striking and a pretty grim picture, because we would prefer river distance to come out as the strongest, not flow patterns. The reality is that's not the case. So we are having a direct impact on a species in a very short period of time. So, flow alteration is having an impact at a genomic level. The current trajectory is pretty concerning, particularly in the Sierras. The Sierra populations in general have lower diversity than the coast already, and then add that layer of additional sort of isolation and limited connectivity, and the trajectory is pretty concerning. So, as some of the options that are ongoing, flow management is one. Myself and others have suggested potentially flow management for recession limbs, which would help the species in avoiding those pulse flows during certain times of the year. But in addition, I really think that listing some of these populations as distinct population segments, or in some way listing them, may afford some protection. The reality is it might be too late. And the other thing I do want to say is this is a really powerful method, and it's really effective. We can use this for fish, for frogs, for bugs, for a whole different slew of different things, and it could be a really powerful monitoring tool, not just single assessment, but because we can detect changes fairly rapidly. You can imagine doing this every 5 years in a monitoring program to actually see if the benefit of a new flow regime or a new management technique is having an effect on increasing connectivity or something like that. Thank you. That's it. Any questions? One question? Yeah. Go ahead. Oh.
Do you think non-native fish predation might have any impacts on the frog populations in the unregulated versus regulated?
Yes, I think invasive species certainly have an effect, but the reality is I've seen them in both systems. So we see crayfish, bullfrogs, smallmouth bass across the board in all these systems. But I'm certain that's probably having some sort of impact, yeah. Thanks.
All right. Thanks.